Welcome to the lecture on the tricarboxylic acid cycle, the TCA cycle. This is going to be the metabolic hub of reactions in the cell that will link catabolic reactions with electron transport chain and oxidative phosphorylation. Lots of material, lots of detail in this lecture, so the lecture objectives will help guide your study and help you focus on what's important and the level to which you need to understand the material. So acetyl-CoA, we've talked about before, is a common intermediate for catabolic reactions. Glycolysis makes pyruvate, which can be converted to acetyl-CoA. Fatty acids, when they're oxidized, will lead to the direct production of acetyl-CoA. And then amino acids, some of them will decay to glycolytic intermediates, but some will go to acetyl-CoA, and we'll see that later. And what can you do with that acetyl-CoA that you make? Well, the primary use of acetyl-CoA is going to be the TCA cycle, which will extract electrons from acetyl-CoA, set up the reduced cofactors that are needed for the electron transport chain, and lead to oxidative phosphorylation. Other things that acetyl-CoA can do is it can be converted to those molecules called ketone bodies, which is a way of repackaging the acetyl-CoA into a stable form that can be released into the blood, and then other tissues can pick those ketone bodies up and use them for energy. And then acetyl-CoA is a pretty important starting material for the synthesis of fatty acids, but also the synthesis of cholesterol. And we'll deal with these in subsequent lectures, ketone bodies and sterols. But today we're going to talk about tricarboxylic acid, or TCA cycle. The TCA cycle is considered to be the hub of biochemical metabolism in the cell because it relates the catabolic reactions to electron transport chain and ATP production, as I mentioned before. So its function then is to catalyze the oxidation of acetyl-CoA, and its products are going to be CO2 and water, but the really important products are going to be electrons that are extracted from that acetyl-CoA. And again, electrons aren't free in solution. They're going to be carried by NADH and FADH2. They're the primary electron shuttles that carry the acetyl-CoA electrons into the electron transport chain. So the goal of the TCA cycle then is to conserve the energy of acetyl-CoA oxidation. The energy contained within those electrons, you want to conserve that, and you want to transfer that energy to the electron transport chain so that that energy can be used to set up the proton gradient that will lead to ATP synthesis. And the way to do that is to use these reduced cofactors as electron carriers to transfer the electrons and their energy to the electron transport chain, which is here. In the process, you take an oxidized NAD or an oxidized FAD. In the TCA cycle, when they receive their electrons, they become the reduced NADH, they become the reduced FADH2. These molecules will then interact with the electron transport chain and give the electrons away. And so the reduced NADH gives away its electrons and it becomes the oxidized NAD, and FADH2 gives away its electrons and it becomes the oxidized FAD. And that completes the cycle. Now new electrons can go into NAD and FAD to reduce them and then transfer them to the electron transport chain. In the process, a proton gradient is going to be created by the electron transport chain, and the gradient is set up using the energy contained within those electrons. And the proton gradient has energy. If you remember from the membrane transport lecture, you use the energy of the proton gradient as the energy source needed to drive the synthesis of ATP by the ATP synthase. So we consider the outcome of the TCA cycle in terms of what is produced for each complete cycle. And we know that 2 carbons will enter the cycle as the acetyl group. And here's the acetyl group. So you see 2 carbons, a methyl group, and a C double bond O. And we know that 2 carbons are released as carbon dioxide. We'll see a little bit more detail on this in a few more slides here. And so we say 2 in and 2 out. That means there's no net synthesis of carbon content, no increase in the total carbon of the cell due to the TCA cycle. If you've had biochemistry before and you've looked at how plants use the TCA cycle, you know that they do increase their carbon content. They're trying to synthesize glucose. We're not going to go there. We're not going to be concerned about that. We're going to be interested in human metabolism. And in the case, we have 2 carbons in and 2 CO2s out. And then for each turn, each complete cycle that we have, we're going to produce 3 reduced NADH, 1 reduced FADH2, and 1 high-energy triphosphate, which is a guanosine triphosphate. We'll see that detail on a couple of slides coming up. And then we mentioned before that these reduced cofactors will then interact with the electron transport chain and lead to the production of ATP. So during oxidative phosphorylation, which is the function of the electron transport chain, and then ATP synthesis by the ATP synthase, we know that 2.5 moles of ATP are produced for every mole of NADH. We know that 1.5 moles of ATP are produced for every 1 mole of FADH2. And if you think about the fact that there are 3 NADHs produced for each turn of the cycle, and there is 1 FADH produced for every turn of the cycle, 3 times 2.5 plus 1 times 1.5 is equal to 9 molecules of ATP produced for every turn of the TCA cycle. And we get that 1 extra GTP molecule being produced. And so we say 10 high-energy phosphate molecules are produced for each turn of the TCA cycle. So here are the reactions of the TCA cycle, and if you've had biochemistry before, you were most likely asked to memorize the molecules, the enzymes, the structures, the electrons, the reactions, and that's not a horrible goal if you think about a master's level biochemistry class. But for this class, we're really interested in the logic of the TCA cycle and the function and the role of the TCA cycle for the cell to do what is needed to meet its energy needs. So I would rather focus on the understanding, given a picture of the TCA cycle, to be able to recognize the elements and understand how they work together. And so the level of this picture here, if you can tell the story of this picture, you're going to be in pretty good shape for being able to answer questions about this picture. So there's 8 steps altogether. There are condensation reactions where things come together, isomerizations, changes in structure. We have oxidative decarboxylation reactions. That means simply that electrons are being removed from a molecule, that's oxidation, but you're also losing a CO2 from a molecule, that's a decarboxylation. Dehydrogenation is where you remove a hydride, 2 electrons and a proton. And then there's also hydrations, which is the addition of water, HOH, across the double bond. There are very specific enzymes for every step, and there are cofactors that are being used to help with these reactions. And we're also going to see coupled reactions, and we've seen this already in the bioenergetics lecture. You put 2 reactions together that happen at one time, and things that are endergonic, that need energy, can be coupled to a reaction that is exergonic, that liberates energy, and you can make these reactions go in a spontaneous way. And again, this is reminding you that 2 carbons come in and 2 carbons go out. And there are a number of cofactors that are going to be very important for TCA cycle function. So we have thymine pyrophosphate, we have the flavin adenine dinucleotide or the flavin mononucleotide, we have nicotinamide adenine dinucleotide, the reduced form or oxidized form, coenzyme A, lipoic acid, and of course minerals like iron, magnesium, sulfur, phosphorus. There are others. And so I always put this note on here. If anybody's looking to increase their energy metabolism, then it's important to increase the dietary intake of the B vitamins and of course these minerals. These things will help, and it always helps to get them from whole natural sources of food. So if we just go through the reactions here, and again the focus is on the story. Tell the story. You know, I love to put these pictures on a test question and ask a question about it. What's going on? What happens if something's wrong or whatever like that. So as we mentioned, acetyl-CoA is the star. The TCA cycle's function is to oxidize acetyl-CoA all the way down to CO2, water, and reduced cofactors. So acetyl-CoA will enter the TCA cycle by reacting with the molecule oxaloacetate. A 2-carbon molecule comes together with a 4-carbon molecule. This is a condensation reaction, and you get a 6-carbon molecule here called citrate. Notice that the acetyl-CoA is being carried by that cofactor CoA here that gets displaced, and the oxaloacetate takes its place right there. It's its position, how it's attached together. Now again, the chemistry is pretty complicated. We'll see this in a couple of slides. You can't just look at this and immediately know which atom is attaching and what the rearrangements and where the electrons are. It's extremely complicated. There's many enzymes that work together for each one of these steps in kind of like a complex, and bring about the changes that you see for each of the reactions. Okay, so a citrate is formed. The next step is to do an isomerization reaction where you change the citrate to isocitrate. And the main point that you notice is right here, there is an OH at position number 3 here, and you want that OH now to be at position number 4, and that's what happens. And there's a reason for that. If there's a hydrogen covalently bonded to this carbon here, this carboxyl acid here is going to pull the electrons. Those oxygens are electron withdrawing, and they'll pull the electrons in that direction, which will partially unshield this carbon, and that will unshield this proton and make it a very attractive delta positive that's going to be the site of the next reaction where electrons are going to be removed from this molecule. So you do a rearrangement to make the next step chemically more straightforward. And that's exactly what happens. And the next step here, here's isocitrate, and we have this oxidized NAD, and it's going to extract the hydride that's at this position right here. And so you'll lose the proton and the 2 electrons, and that hydride goes on and becomes NADH. That's our first cofactor, our first reduced cofactor that's formed. In the process, this CO2 is going to be removed as well. It's going to be liberated as a carbon dioxide. That's a waste product. And in the end, you're going to be left over with the CH2, where there used to be the hydrogen and the carboxylic group. Again, the reaction is complicated. You'll see this in a couple more slides here. So the next step is very similar. You're going to do a reaction that is going to lead to the oxidation of alpha-ketoglutarate to form succinyl-CoA. You see the NAD is here. That is the oxidized form, but you see it's leaving as NADH. So you know a hydride has been transferred off of the substrate and onto the product NADH. You know this is an oxidative reaction because of that. And in the process, you're also losing this CO2 down here on the end. So that's your second carbon that goes away. But again, it's almost impossible when you look at this simple reaction here. Well, what are the electrons that are changing, and what are the bonds, and where's the attack, and what's going on, and where does the coenzyme make? That's why we don't go through the reaction mechanism per se for each one of these steps. But it is sufficient to know that you have your second oxidative decarboxylation that leads to the formation of succinyl-CoA. And this coenzyme A now comes in, and it's going to attach to this small molecule here. It's going to serve as a carrier. Okay, so let's look at this reaction. Let me show you why we're not going into a lot of detail for each one of these reactions. There's 2 things to point out here, and that is the complexity of the decarboxylation reaction, but also the critical role that these cofactors play in this process. So you start with an alpha-keto acid. This could be isocitrate, or it could be alpha-ketoglutarate. Those are both alpha-keto acids, and the 2 enzymes, isocitrate dehydrogenase and alpha-ketoglutarate dehydrogenase, are both doing oxidative decarboxylation reactions here. So when the alpha-keto acid comes along, the first thing we want to do is we want to eliminate this carboxyl group. We want to eliminate it as CO2. And a great way to do that is to use a dehydrogenase enzyme that has the cofactor thiamine pyrophosphate, TPP. And if you remember the cofactor lecture, TPP is a very strong nucleophile that can lead to decarboxylation in reactions. That was what that cofactor does. And in fact, the TPP acts as a nucleophile. It hits that carbonyl carbon right there because of the delta positive, and now the TPP is connected to that carbon that we saw before, and the leaving group is the CO2. So you've achieved goal number 1, and goal number 1 is to eliminate a CO2. Okay, that's fine. But now you have a problem. This substrate molecule is now covalently bound to the active site of this enzyme, and that's not what's supposed to happen at all. You need to release that product so it can go forward in the TCA cycle. Well, how can you do that? Well, we need another molecule here who is a stronger nucleophile that has the ability to attack this group and transfer that group off of the TPP. And our friend lipoic acid is going to be exactly what we need here. This enzyme is called transacetylase, and In this case, the lipoic acid is in the oxidized form. If you remember, lipoic acid has two roles. It can serve as an oxidation reduction cofactor, but it can also serve as an ACO carrier. And we're going to see both of these roles here. If you remember, the disulfide bond is the oxidized form of lipoic acid, whereas the two independent SH groups or SR groups are the reduced form. So when lipoic acid attacks this TPP, you get a transfer of the substrate onto one of these sulfur groups, as you see there. And in the process, electrons are extracted from this molecule and given to break the disulfide bond, to give the electrons necessary so that you have 8 electrons around each one of those sulfurs. And so in this case, you see the alcohol, the OH, which is more reduced, is becoming a C double bond O, a carbonyl, and that's going to be more oxidized. And that's because 2 net electrons have been extracted from this molecule in order to reduce that disulfide bond. Okay, so now you've done something interesting. You've got electrons repositioned inside this molecule now. So far, I don't see the production of any reduced cofactor, and I sure don't see the product molecule coming off yet. So we have a little bit more work to do. How are we going to get this current product molecule off of the transacetylase? The way to do that is we bring our friend coenzyme A, which is also a cofactor, and coenzyme A has that sulfur, which acts as a nucleophile, and that nucleophile can attack this carbonyl carbon, and in so doing, it's going to break the bond between the lipoic acid sulfur, and it will attach that product molecule now to the coenzyme A. And there you have your product. In this case, it's either alpha-ketoglutarate, if you're doing the alpha-ketoglutarate, the isocitrate dehydrogenase, or it's going to be succinyl-CoA if it's the alpha-ketoglutarate dehydrogenase. Okay, so now you've created your product molecule. That's fine, so that molecule can now go forward in the TCA cycle with the upcoming reactions. But we now have another problem we didn't have before. We've pulled that product molecule off of the sulfur on the lipoic acid, but the lipoic acid now is still in a fully reduced form. And for this lipoic acid to do what it needs to do, it has to be in the oxidized form so it can pick up this intermediate molecule and go forward. So the first thing we need to do is oxidize that lipoic acid to get it back in that disulfide form. Well, here comes a molecule of flavin-containing enzyme. This is FAD. You see it's oxidized because it doesn't have that H2 on it. And when the FAD, through this enzyme E3, works together with the transacetylase, you're going to extract electrons from these two SH groups. That's going to oxidize the SH groups and give you that oxidized form. And then the FAD becomes the fully reduced FADH2 form here on that flavoprotein. So now we've moved electrons off of the intermediates, but they're still not in a mobile form yet. And then our friend NAD, the oxidized cofactor, will come and bind to this enzyme. It will extract those 2 electrons away from the FAD, oxidizing it back to FAD here. FADH2 becomes FAD, and the oxidized NAD will become NADH. And now NADH is that soluble mobile carrier, and it can move to the electron transport chain and give off the electrons. So that was a lot of detail, but I really wanted to show you that if you see a reaction, you see 2 molecules becoming one or the other, there's a lot more to it that's not shown on those really simple 8 reactions as you go around the cycle. So if we keep going now, succinyl-CoA is going to be converted to succinate, and look what happens along the way here. We're going to take a GDP, a diphosphate molecule, plus an inorganic phosphate. These are going to bind to that enzyme. And in the process, this squiggly line here says that this is a high energy bond, but it's also labile, which means it's easily broken. And so you break this bond between the coenzyme A and the succinate molecule, or what's going to become the succinate molecule, and that energy is used to pay the energy cost of phosphorylating GDP to make the higher energy GTP molecule. And so this is an example of substrate level phosphorylation where you create a nucleotide triphosphate molecule in solution without molecular oxygen. And of course in the process when you break that coenzyme A bond, you have free coenzyme A, and then the product over here is free succinate. Then we go forward here, succinate gets converted to fumarate, and if you notice, succinate has all single bonds. We call this a fully saturated molecule, at least for the 4 carbons in that sequence there. And when you form fumarate, you have introduced a double bond. And anytime you go from a single bond to a double bond, you know electrons have been lost. This molecule is oxidized. Succinate is oxidized to fumarate. Where do the electrons go? Well, succinate dehydrogenase is an enzyme that contains the flavin, FAD, and in its oxidized form, it can receive those electrons. So the electrons are transferred from succinate onto FAD. That creates the reduced FADH2, and in the process, you produce the oxidized fumarate here. Fumarate then will go forward to malate with a very normal hydration reaction here where you add HOH across the double bond, and the OH of the water ends up on this carbon and the H here. If you notice there's symmetry, there's a carboxylic group, a carbon, so there's really, you can't say who's number 1 or 2, there's symmetry. And so you end up with an OH on one of these carbons and an H on the other. And whenever you have an OH next to a hydrogen covalent bond, this OH is going to be electron withdrawing, and that's going to leave that hydrogen a little bit unshielded. It's a nice delta positive. And our friend, the oxidized NAD, can come and bound to the enzyme. It can extract that hydride, the proton and the 2 electrons. You remove that entire covalent bond and you create NADH, which is the reduced cofactor, and in the process you have created a C double bond O, the more oxidized form of carbon, which is oxaloacetate. And we're back at the top now with the next acetyl-CoA that will come in. So we mentioned this previously in the bioenergetics lecture, so you get to see this again. Having seen the TCA cycle, maybe it'll make a little bit more sense to you. The cycle is about 90% efficient in terms of conserving the energy that you get. Each of these electrons are being pulled out from the acetyl-CoA. You want to save that energy, and so it's very efficiently transferred to the reduced cofactors. Kind of neat thing about our bodies and how they work. And if you recall, there are several reactions here that have large negative delta Gs. And what the negative delta G says, this is an exergonic reaction. It means it liberates free energy, and because energy is liberated, it's considered spontaneously as written. So if you have oxaloacetate and acetyl-CoA as the substrate, the reactants, and citrate as the product, the negative 7.7 says that this equilibrium will lie to the right. It will favor the formation of citrate. Isocitrate will favor the formation of alpha-ketoglutarate, and alpha-ketoglutarate will favor the formation of succinyl-CoA. Okay. Now, because of these large negative delta G values, these things are considered thermodynamically irreversible. You can drive these reactions forward because they are spontaneous or favorable due to their negative delta G, but it's very, very difficult to get these reactions to go backward because that's a lot of energy you have to pump in, and there's really no reactions around this with sufficient energy to drive them backwards. So we call them thermodynamically irreversible. There's a couple of steps here that are endergonic. Endergonic means that you need energy to be put in. You have to put energy into the reaction to make it go, and that is citrate to isocitrate, and it's also malate to oxaloacetate. And as you see here, the lines tell you a little bit about it. Because of the positive delta G, you see you have a large arrow going backwards and a small arrow going forward. So that shows you that you favor the reactants over the products. And the same thing is over here. You have a lighter arrow going to oxaloacetate, and you have a stronger arrow going back to malate because you are favoring the reactants here. And that's going to be pretty important. If you remember, if you couple a negative delta G reaction, an exergonic reaction, to an endergonic reaction, it's possible to draw the reactions through to the final product over here. So if you have an oxaloacetate, if an acetyl-CoA comes in and it combines with oxaloacetate to make citrate, you are depleting the amount of oxaloacetate that you have at any given time. This reaction has an equilibrium constant, and that equilibrium constant is based on the ratio of these two molecules. So if you consume oxaloacetate, you perturb that ratio, and so malate then will be driven to oxaloacetate to help get that ratio back to where the equilibrium constant says it needs to be. And meanwhile, acetyl-CoA will draw more of it over to citrate, and malate will come back over. So you see by coupling these things together, you can get these, and the same thing is true over here for the citrate to isocitrate. You can draw isocitrate around to alpha-ketoglutarate. So the key thing for us now is going to be you need to think of a way to regulate these enzymes, the ones that have large negative delta Gs that are spontaneous and will lie heavily to the right. You've got to make sure you really want to have those products, and if you don't, you need to regulate those enzymes. And likewise, if you need these positive delta Gs, these inorganic reactions to go forward, then you need to do something to the product molecules to favor movement of the substrate to the product through those inorganic steps. This leads us to the idea of efflux intermediates from the TCA cycle. And efflux is just a word that says to leave or to go away from. And our TCA cycle really is the metabolic hub of metabolism in our cells, cells that have mitochondria. And if your cell has lots and lots of energy, if it's really feeling full of resources and there's not a lot of exertion going on, well, the TCA cycle activity is just not needed. There'll be some need always. Of course, the TCA cycle reactions are going to go on to keep the energy balance up. But if there comes a time when you have excess energy and you just don't need a lot of TCA cycle activity, it's possible for a number of the intermediates to leave the cycle and go do something else. Now, it doesn't mean that they pack their bags and jump in their car and drive away. That's not at all what it means. Rather, there is a pool or a collection or a supply of molecules in the cell. There is a supply of acetyl-CoA. There's a supply of citrate, a supply of alpha-ketoglutarate, and so forth. And at any given time, if the TCA cycle says, I want to take citrate and change it to isocitrate, it's just going to grab one of the citrates that's sitting there in the mitochondrial matrix, and it'll be converted to isocitrate. But if the TCA cycle is not operating and you're not consuming citrate to isocitrate, that pool will still sit there, and some other enzyme can grab that citrate and say, why don't you come over here? I need you for my reaction. And you can turn it into a fatty acid, or you can turn it into a cholesterol molecule. And the same thing is true for alpha-ketoglutarate. If the energy level is high in the cell and you don't need a lot of TCA cycle, the alpha-ketoglutarate level will increase inside the mitochondria. And there are other reactions that will grab that alpha-ketoglutarate and convert it into some other molecule that's necessary. Typically, glutamate is one amino acid that's formed, and then that can be transformed. Or you can grab the alpha-ketoglutarate and lead to a variety of neurotransmitters that can be synthesized from that. And that's the idea. During times when you're energy rich, and you don't need all those intermediates for the TCA cycle, you can use those intermediates for other reactions. We'll cover those reactions in a lot of lectures coming up. We also have reactions that can bring intermediates back into the TCA cycle, and they're called anaplerotic reactions. Anaplerotic is a word that means filling up or to restore. And so in this case, when the TCA cycle feels a need to become more active, when the activity is required, you want to bring molecules in that will repopulate these intermediates so that the TCA cycle can resume normal activity. And there are two main anaplerotic reactions that we'll see quite a bit. One of them is going to be pyruvate, which will become oxaloacetate, and the other is glutamate, which will become alpha-ketoglutarate. Although you could get amino acids making some other intermediates as well. But that will refill these pools and allow the TCA cycle to start running again. So let's look at that first one here. The first one here is pyruvate to oxaloacetate using the enzyme pyruvate carboxylase. Pyruvate carboxylase is going to be an enzyme that allows pyruvate to enter the TCA cycle by going directly to oxaloacetate. Instead of the other pathway you've seen before, pyruvate goes to acetyl-CoA, and then acetyl-CoA will enter the TCA cycle by combining with oxaloacetate. So why would that be important? It's important because during times of catabolic reactions, these produce a lot of acetyl-CoA. And if you have a lot of acetyl-CoA, then that would suggest you need more oxaloacetate that could combine with that acetyl-CoA that could lead to citrate and drive the TCA cycle forward. So this anaplerotic reaction, this pyruvate carboxylase says, well, you have enough acetyl-CoA. I don't want to send my pyruvate to acetyl-CoA and just add to this huge excess you have. Rather, pyruvate carboxylase becomes active, to convert the pyruvate to oxaloacetate. Pyruvate is a 3-carbon molecule. Oxaloacetate is a 4-carbon molecule. So in this case, you're going to add a CO2 onto the molecule to give it 4 carbons. It's going to take energy to do that because you're increasing the energy status of the molecule, and you're going to hydrolyze an ATP molecule. You're going to liberate that amount of energy that comes from just breaking that phosphate anhydride bond, and that energy is what is used to drive the formation of oxaloacetate. And it doesn't say on here, but the cofactor involved is biotin. And if you remember biotin, that is the cofactor that's involved in carboxylation reactions. And that's really an important step that we'll see a couple of times when you need to fill up your supply of oxaloacetate to get a lot of TCA cycle activity. We mentioned a few slides ago that reactions that have a large negative delta G need to have enzymes that are very carefully regulated. If you have a large negative delta G, the spontaneity of that reaction suggests that you will get a lot of product formation. But the TCA cycle doesn't always need to be running, and you don't need a lot of product formation. If a cell is at rest, if it has a lot of energy stored up, if its energy needs are met, then you want to downregulate the TCA cycle so you can conserve that energy until it's needed. And so the reactions that have large negative delta Gs, the exergonic reactions you see, have a number of negative allosteric modulators that are present. For example, if the TCA cycle activity is not needed, citrate can serve as a feedback allosteric modulator, and that citrate will say, stop, I don't want any more of this acetyl-CoA coming in. You can use the acetyl-CoA for something else. You don't need to come into the TCA cycle. Or over here for the NADH producing reactions, 1, 2, 3, NADH, if the NADH levels are high in the mitochondria, if your energy levels are high, your ATP levels are high, you don't need to use a lot of NADH for electron transport chain or ATP synthesis. And so the NADH inside the mitochondrial matrix will get a higher concentration. And that higher concentration will allow the NADH to bind to the allosteric binding sites, and it happens to be a negative allosteric modulator. And this makes a lot of sense. These are the NADH producing steps. And if NADH levels are high, you don't need to produce more NADH. And so NADH becomes a really great signal to say, no, I don't need you to be active. I'm going to allosterically inhibit you. Now let's look over here, for example. If you remember malate dehydrogenase, this had a largely positive delta G. And that would suggest that you'll have more malate and less oxaloacetate because of that positive delta G. But we mentioned before that when acetyl-CoA comes in and it combines with oxaloacetate, it draws that malate across to oxaloacetate, and then acetyl-CoA and oxaloacetate becomes citrate. So you will be drawing these reactions through to citrate, so it's important to regulate this malate dehydrogenase enzyme as well. And so NADH becomes a powerful signal that says, don't make more NADH, I've got plenty. Now what happens when a cell is using energy, when it's doing work, or muscles are contracting or nerves are conducting or whatever? During times that the TCA activity is high, there's going to be a need for NADH. NADH will be used by the electron transport chain, and then the ATP synthesis will go on. So the NADH will be consumed by the electron transport chain. The concentration of NADH will be lower inside the mitochondrial matrix, and when the concentration of NADH is low, there's not enough NADH to bind to these enzymes and allosterically inhibit them, so you lose the allosteric inhibition. And these enzymes now become more active, and you'll clear metabolites through these steps much quicker. So NADH is a very powerful signal that controls the TCA cycle. When you're at rest, when NADH levels are high, you don't need TCA cycle activity, and these 3 enzymes are going to be downregulated. At times when energy production is very high and you need a lot of NADH, then these enzymes will not be inhibited. They will produce their NADH and they'll be fully active. Now there are other signals here. You see ADP and calcium are both green pluses. That means they are allosteric activators. They are enhanced by ADP and they're enhanced by calcium. So let's think just for a moment, why would ADP be a positive allosteric modulator for this enzyme, isocitrate dehydrogenase? Why would that be? Well, what is the function of the TCA cycle? The TCA cycle's real job is to extract those electrons, put them on those reduced cofactors, and send them to the electron transport chain so you can make more ATP. And if your ATP levels are low, that suggests that your ADP levels are probably higher. And so if ADP is high, it becomes a pretty important signal that you need to make more ATP. And that would explain why ADP is a strong positive allosteric modulator of this first step here. Now, I'm oftentimes asked, why wouldn't you have ADP on this step, because it's important there too? Well, if you remember, citrate to isocitrate is a positive delta G reaction, so it's going to favor citrate right here. If you can activate the isocitrate dehydrogenase reaction, you can pull isocitrate around to alpha-ketoglutarate, then the negative delta G will pull you on through to succinyl-CoA. So ADP will turn on the isocitrate to alpha-ketoglutarate, and then the reaction will just tumble on through. So in a way, ADP can affect one enzyme, but it can affect flux through both enzymes. Now, what about calcium? Why would calcium have an effect here? We really haven't talked much about calcium, but we will talk about calcium more and more. Calcium is a really important ion. Typically, the calcium concentration inside a cell is low, and calcium is very high outside the cell. And so when calcium comes into the cytoplasm, it's a powerful signal that tells the cell it's time to do something. If you're a muscle cell, calcium will stimulate muscle contraction. If you're a nerve cell, calcium will stimulate conduction of electrical information. If you're a cell that exports molecules, then calcium will signal that vesicle migration to the membrane and fusion so that you can release whatever the molecule is you want out. So calcium is a signal that it's time to do the work of the cell. And if that's the case, then that calcium signal will communicate with the TCA cycle and say, I need you to become more active. You need to produce more electrons and electron cofactors here so that we can do more oxidative phosphorylation and make more ATP. So we now see three signals, really two key signals, but three signals overall, depending on the cell type. NADH is the most powerful because it is going to be the primary signal that tells the cycle to be more active or less active. And then ADP is going to be important because it signals, if ADP is high, it signals that ATP is a little bit lower. And then for cells that are excitable or do mechanical work, then calcium becomes an important signal saying that it's time to do the work. And here's a note down here. We'll talk about pyruvate dehydrogenase in a few minutes, but it's also regulated by NADH. That pyruvate hydroxylase enzyme sits right here, and it will actually control the production of acetyl-CoA, and it is also under the signal, under the NADH signal as well. So let's think about overall regulation of metabolic pathways, and we can think about the TCA cycle. There's going to be rate-limiting steps. That means steps that an enzyme can be more active or less active based on whatever allosteric modulator it has. We've talked about committed steps where you have a reaction like when glucose gets phosphorylated, it's trapped inside the cell. But when the glucose 6-phosphate becomes fructose 6-phosphate, and then you get the fructose 1,6-bisphosphate, that's the committed step for glycolysis. So you see committed steps can be involved in regulation. We have thermodynamically irreversible reactions where the delta G is so largely negative that you really can't go backwards. We've seen feedback inhibition. We just saw product inhibition with citrate affecting the formation of citrate, as a feedback inhibitor, but also we see NADH, which is produced at several steps in the TCA cycle, then affecting several other steps in the TCA cycle, so that's feedback. And then we saw compartmentation in the earlier lecture, and we're going to see regulation coming up. So a couple of pathways, just general pathway regulation strategies we have. So anytime you have a rate-limiting enzyme, the idea is pretty much in the words here. It's an enzyme that can allow more reaction to happen or less reaction to happen. And it's kind of like construction on a highway, as you know. You're on a highway and all the cars are going along and there's some kind of construction where they've closed a couple of lanes. And that's going to cause all of the traffic to back up for however long it goes back here. And because there's only one lane open here, only one of these cars or only a couple of molecules can slip through per unit time. The actual flux or movement of molecules is greatly restricted, and because that rate of flux is lower, that means the enzyme is limiting the overall rate of the pathway. But you know, if somebody came along and said, okay, we're done with the construction here, we're going to remove this barrier. Once that barrier goes away and all three lanes are open, then you increase the flux. There's a great amount of flux or movement of molecules through that step, and you can clear them through a lot faster. Okay, so that would be whether you impose the rate limitation or remove the rate limitation. Now, what's interesting about these efflux reactions we talked about before, if this barrier is in place and all this traffic is backed up and only a few of these molecules or cars can slip through, these cars are sitting here going, well, I'm not going to sit here in this traffic mess. I see an exit over here, and so I'm just going to leave and go find a different pathway where I can get around and do what I want to do. And this is a great example of an efflux pathway where you're taking molecules that are not needed and you're just letting them go off and do something that is needed. And then, of course, if you remove that barrier and things start to move, you're more likely to send the molecules straight along the path, and you're less likely to send molecules out through the efflux pathway. Okay, so let's look again at acetyl-CoA and pyruvate, how these things work together. So if you recall, acetyl-CoA is going to be a common intermediate for these catabolic reactions here. If you break down fat, you make a lot of acetyl-CoA. When you break down protein and you oxidize those amino acids, a lot of the products end up as acetyl-CoA. And of course, glycogen, glucose, pyruvate, and acetyl-CoA. So that's the common intermediate. But pyruvate now, pyruvate which is produced by glycolysis, pyruvate has multiple fates in the cell. Pyruvate normally will go to the TCA cycle through this by forming acetyl-CoA, as we saw before there. That's really the main pathway for acetyl-CoA. Acetyl-CoA joins with oxaloacetate, you get citrate, and into the TCA cycle you go. If there's a lot of acetyl-CoA already present and you need more TCA cycle activity, we mentioned before that this pyruvate can be converted to oxaloacetate right here through pyruvate carboxylase. And so you can fill up the oxaloacetate with more molecules to add to the fact that you have a lot of acetyl-CoA, and those two can combine and they can move you forward. But if that's not needed, or if you don't have mitochondria at all, this pyruvate doesn't really want to go by oxaloacetate or by acetyl-CoA. Under those conditions, you convert it to lactate using lactate dehydrogenase. And lactate is going to be the molecule that gets released from the cell into the blood, and then some tissue that can use lactate will pick it up. That's the case where you don't have mitochondria, or that's a place where your TCA cycle is running so slowly that you don't need oxaloacetate or acetyl-CoA. And then the last thing you can do is you can say, well, pyruvate can be transaminated. You can go through a reaction that takes that 3-carbon skeleton and turns it into an amino acid. And then alanine can be used for a variety of processes. Protein synthesis or some other reactions that take place. Okay, so these two things are going to work together. If you are currently using glucose and fats as energy sources, you're going to be producing an awful lot of acetyl-CoA, and the TCA cycle is going to be filled up with acetyl-CoA. But under times where maybe you haven't had carbohydrates in your meal, or the blood sugar is a little bit lower and you need to burn more fat, your acetyl-CoA will come primarily from beta oxidation, and that's going to affect the ratio of acetyl-CoA to oxaloacetate. We will build those arguments a little bit better coming up in the fat lecture. Okay, let's look at pyruvate dehydrogenase, which is really the main reaction by which pyruvate becomes acetyl-CoA and the acetyl-CoA enters the TCA cycle. Under conditions like if a person is exercising or they're doing a lot of energy expenditure and so their cells need to be making a lot of ATP. Under those conditions, the TCA cycle is running at full capacity. The pyruvate that's produced by glycolysis will become acetyl-CoA, and the acetyl-CoA will enter the TCA cycle. And the enzyme that does that, PDH, pyruvate dehydrogenase. And you see that it's going to convert the 3-carbon pyruvate to the 2-carbon acetyl group, and that 2-carbon acetyl group gets attached to coenzyme A. So what goes in? Pyruvate, free coenzyme A, and in the process, this is also an oxidative reaction. We see that oxidized NAD goes into the enzyme and it comes out as a reduced NADH. NADH. So electrons are being extracted from pyruvate as it becomes acetyl-CoA. And then that NADH just goes on over to the electron transport chain and gives the electrons away. So again, this is an NADH producing reaction. So if the NADH level inside the mitochondria is high, you don't need to make more NADH. NADH becomes a negative allosteric modulator of this PDH enzyme. It will shut it down. Or if you have a lot of NAD, oxidized NAD, that's going to be a signal saying, hey, I've used up all my NADH, I need more. And NAD becomes a positive allosteric modulator. If the cell is doing a lot of glycolysis, and typically for skeletal muscles, when they're exerting a lot of energy, they will increase their glycolytic activity. Those cells will make a lot of pyruvate, and as pyruvate starts to build up, pyruvate is also a positive allosteric modulator. It says, I need to move the pyruvate through to acetyl-CoA to get the TCA cycle going so I can get my electrons, so I can do oxidative phosphorylation by the electron transport chain and the ATP synthase. So this over here is a little bit more confusing, but if you recall, under the metabolic hormones of insulin and glucagon, glucagon is going to lead to the phosphorylation of some proteins, and insulin is going to lead to the dephosphorylation of those proteins. They work together. So during times when the TCA cycle is not needed, or during times when the cell says, you know what, I've got plenty of energy, I don't need more, you will have a hormonal signal, you'll have a glucagon response where this enzyme, PDH, will become phosphorylated and it becomes an inactive enzyme. So there's the phosphate that's been added. And the enzyme that does that is the PDH kinase. It is a kinase enzyme that gets activated by the glucagon signal, and it leads to the phosphorylation of the enzyme. And this is showing you that ATP is used by the kinase, and that ATP is transferring a phosphate onto the enzyme to give you that inactivated PDH enzyme right there. Okay. During times when you want more energy, like under an insulin signal, where insulin is saying we've got more than enough glucose in the blood, bring that glucose in, use it for energy, use that energy to build molecules, build cells, build proteins, all these kind of stuff, then you will want to dephosphorylate that PDH, and you want to bring it back over to the active form. And so here you see the PDH phosphatase, where that phosphate bond is broken and inorganic phosphate is liberated. Now, what are these green signals right here showing you? The green signals are showing you the signals that regulate the kinase and regulate the phosphatase. In this case, if the cell is saying, I don't need my PDH to be active, and so it gets phosphorylated by the kinase, that's fine. But if your ADP levels are high, ADP says, now wait a minute, ATP levels are lower, I need to get more TCA cycle activity, I need to get more electron transport chain, I need to get more oxidative phosphorylation. And so ADP becomes a very strong negative allosteric modulator of the PDH kinase. So you'll stop the phosphorylation reaction and you'll keep this enzyme in the active form. And likewise, calcium is that signal that says, you know, something exciting is happening. We're going to start doing work. We're going to contract or conduct or, you know, send substances out or whatever. So calcium is going to tell this PDH, I need you to be active. And so calcium will stimulate the PDH phosphatase, and that phosphatase will dephosphorylate the PDH and bring you back over to the active form. And so you have glucagon and insulin, which are trying to phosphorylate and turn off or dephosphorylate and turn on. Those are the metabolic hormones. But then you have the allosteric modulators, ADP and calcium, that tell you energy is required. Stop inactivating my enzyme, and while you're at it, why don't you activate any unactivated enzymes? And down here, it's just the same figure as up here, just a little bit more detail. It doesn't show you this. Here's 3 carbons becoming 2 carbons, but down here you see that the CO2 comes off. It's actually a decarboxylation reaction, similar to what we saw for alpha-ketoglutarate dehydrogenase and isocitrate dehydrogenase. And you see there's thiamine, lipoate, FAD, and NADH. So the same figure we saw a while back is the same thing that happens for this reaction as well. Okay, just a reminder, we mentioned this before, but if the cell has plenty of energy and you don't really need a lot of TCA cycle activity, then it's important to spare the acetyl-CoA and not send it into the TCA cycle. So under these conditions, if the TCA cycle is not running very well, if you remember, citrate to isocitrate has a positive delta G, which means you'll favor citrate formation. That means you'll have more citrate than normal, and that higher citrate allows this molecule to feed back and become a negative allosteric modulator of the citrate synthase enzyme, shutting it down and preserving the acetyl-CoA to be used for something else in the cell. And we're going to find out that's fatty acid synthesis or cholesterol synthesis in the next couple of lectures coming up. And again, a little bit easier to see this zoomed in. We see that NADH is going to be a powerful negative modulator for the NADH producing steps. The same thing is true for malate dehydrogenase up over on the other side. And then we see ADP and calcium are both stimulatory, getting the cycle to move when energy is required. And the way these things work is just like we've seen before. And so if we take, for example, the isocitrate dehydrogenase enzyme, under normal conditions, the black curve is here. Isocitrate dehydrogenase is a cooperative enzyme, so it has that standard sigmoidal shape. And you know that when the isocitrate concentration is low, the enzyme is, in the T state. It's not very active. It's not very receptive to substrate. But as the isocitrate concentration gets higher and higher and higher, eventually you switch over to the relaxed form. It binds the substrate. Then you get the substrate stimulation of enzyme velocity, and the curve will come up until it reaches Vmax. And there's going to be a KO5 value associated with that. KO5 is that substrate concentration that gives you half maximal activity. Look what happens in the presence of ADP. When ADP shows up, that's the signal that says, wait a minute, we need more energy. We need to get things going here. And so ADP acts as a positive allosteric modulator, which shifts this curve to the left and actually removes the sigmoidal shape. It becomes hyperbolic now, so you almost completely remove the cooperative protein interactions that would give you that sigmoidal shape. And because of that, your half maximal activity is at a much lower level. You see a great increase of enzyme activity at lower levels of isocitrate. And when you see ADP down here, if there's no ADP present, you have a certain amount of enzyme velocity. But as the ADP concentration gets higher and higher and higher and higher, that positive allosteric modulation will give you more and more and more enzyme activity as the ADP level starts to rise. And of course, you can pretty well figure this out, that if your ADP levels are high, you're going to be more over here on the left. But as the TCA cycle gets running and as ATP gets synthesized and the ADP level starts to drop, you'll move back over to the sigmoidal shape, or you'll come back down over here. It's constantly changing. ADP levels rise, TCA cycle activity increases, ATP levels increase, ADP levels decrease, TCA cycle will slow down. So it's really a very finely regulated system. And then we know that NADH is going to be a powerful inhibitor of the three enzymes that produce NADH and pyruvate dehydrogenase, which converts pyruvate to acetyl-CoA. And you see as the NADH concentration gets higher and higher and higher, you very rapidly lose enzyme velocity because that NADH is acting as a negative allosteric inhibitor, which is blocking the enzyme's activity. And then we mentioned malate dehydrogenase before, and that is NADH is produced during this step here. And again, NADH is going to be a strong negative allosteric inhibitor because you don't want to produce more NADH if the NADH levels are high. And there's another good reason for this. When NADH levels are high, even though this has a positive delta G, which will favor malate, if you recall, oxaloacetate will be consumed and it'll pull the reactions through, and this will just make sure that you lock that reaction down if there's high NADH. And then malate is free to be an efflux intermediate. It will leave the TCA cycle and go do something really important, and that something is glucose formation. That'll be when the liver is trying to synthesize glucose to put it out into the blood, and malate is one of the precursors of the materials that's going to lead to that. That'll be covered in a later lecture coming up. Okay, what about the tissues that really don't have mitochondria? Red blood cells don't have mitochondria, so there's no TCA cycle, no TCA activity. Fast twitch skeletal muscle, those glycolytic fibers, they have a very limited mitochondrial capacity. So they can do some mitochondria, some TCA, some oxidative phosphorylation. but primarily they're going to be glycolytic fibers. They're going to use glycolysis. They're going to make pyruvate, and since there's not a lot of mitochondria, their pyruvate is going to be turned into lactate and eliminated from the cell. Cardiac muscle and slow twitch muscle has a lot of mitochondria. These are muscles that are involved in posture and long-term activity with low fatigue, especially cardiac muscle can beat all the time. So because they have a tremendous amount of mitochondria, they can do a lot of oxidative phosphorylation and make a lot of ATP by oxidative methods here. And the brain has lots and lots of mitochondria, and so they have lots of TCA cycle. And you know the brain is going to use glucose as its primary energy source, and so glucose will make pyruvate. Pyruvate gets converted to acetyl-CoA, and then that acetyl-CoA will go into the TCA cycle. So let's put it all together here. Acetyl-CoA is going to be that main entry molecule for the TCA cycle. When I say main, that's to remind you that pyruvate can be converted to oxaloacetate under certain conditions, and so that can bring pyruvate carbons in by an alternate pathway, but really acetyl-CoA is the main entry level. But we know that acetyl-CoA is going to be a very important molecule that will be sensed by the PDH enzyme. If acetyl-CoA levels are high, you won't use that acetyl-CoA to do more TCA cycle. The main products are going to be the electrons that are extracted from that acetate, and they're going to be carried by NADH and FADH2 into the electron transport chain. And then we know the products are going to be CO2, 2 carbons in, 2 carbons out. In this case, the CO2 gets eliminated as waste. Let's look at a sample question where we can get a couple of these comments out here. Despite 2 largely endergonic reaction steps, the TCA cycle proceeds in a forward direction completely through all the reactions of the cycle. Why is this? Wow. Okay, so what are endergonic reaction steps? What does that mean? Endergonic, positive delta G, and there are 2 of them. And we know that positive delta G reactions favor the reactant, not the product. And so the question says, if you're going to get stuck with a reaction that favors the reactant, how can the cycle move forward? So if you recall, the idea overall is you have to take a positive delta G reaction and couple it with a negative delta G reaction. And if the negative delta G if there's more energy liberated than energies that's required for the endergonic reaction, overall those 2 reactions will go forward towards product. And that's what this question is getting at. And in fact, for the TCA cycle, where there is a positive delta G reaction, there is always a nice negative delta G reaction right after it, which can couple together. So you're looking for something that says 2 reactions are coupled together, or the net delta G is negative, something along those lines. Let's see if we can find it. Overall, the net energy change of the TCA cycle is exergonic, negative delta G. Well, that sounds like a good answer. We'll put a post-it note on that one right now. That sounds pretty good. Oxaloacetate will readily react with acetyl-CoA to form citrate, driving the cycle forward. Well, that's true. Oxaloacetate does react with acetyl-CoA, and it does form citrate. It is a negative delta G reaction. But that doesn't tell me why those positive delta G reactions go forward. This just tells me about one of the steps of the TCA cycle. So that sounds like a true answer, but not an answer that answers the question. Here, the equilibrium constant for the endergonic reactions favors product formation. Well, that's just wrong, because you know endergonic positive delta G is going to favor reactant formation. So C is just wrong. The equilibrium constant for the endergonic reactions favors reactant formation. Now that's correct. There you go. Endergonic reactions do favor reactant formation. But there again, it's a true statement, but it does not answer the question. The question says you have these endergonic reactions. How does the cycle move forward? And answer D doesn't answer that at all. And then here is E, the endergonic reactions are coupled to the breakdown of a high-energy phosphate molecule. Well, that might be true for some reactions, but certainly not the reactions in the TCA cycle. Really, we don't see any high-energy phosphate compounds except for when GTP is actually formed, not broken down. So E can't be right. So it looks like A might be our best answer. And in fact, it is. So when the electron transport chain becomes more active, the TCA cycle usually becomes more active also. What is the reason for this? So this question is getting at the idea of the TCA cycle works with the electron transport chain. The electron transport chain works with the ATP synthase, and together they lead to a production of ATP in the presence of oxygen. So if the TCA cycle becomes more active because the electron transport chains become active, what is the reason? Well, this question will be a lot easier to answer after the electron transport chain lecture, which is next. But if you recall, the TCA cycle produces NADH and FADH2. If you recall, the electron transport chain consumes those molecules. It takes the electrons away. So if the electron transport chain is active, it means it's using up all those electrons and all those electron carriers. And the TCA cycle will sense that by saying the NADH level is going down. The FADH level is going down too, but NADH is the main signal. And so as the electron transport chain consumes the NADH, TCA cycle loses the inhibition and it becomes more active. So we're looking for an answer that has something to do with NADH levels and TCA cycle activity. Here the ADP level falls due to increased ATP synthesis. Well, that's true. It's kind of true, but actually, ADP is a positive allosteric modulator, and if the ADP level falls, then it's not going to be a very good binder. It's not going to be enough concentration, so that sounds backwards. The increased AMP level stimulates TCA cycle activity. Nah, if you think about it, AMP was not an allosteric modulator of any of the reactions we saw. Calcium stimulates the TCA cycle. Well, that's true. Calcium does stimulate two of those reactions, but calcium has no effect on the electron transport chain. So obviously that's a TCA cycle thing and not an electron transport chain. Here's NADH level decreases with increased electron transport chain activity. There you go. We saw that before. NADH is a pretty strong modulator of the TCA cycle, and so if the NADH level decreases, the TCA cycle activity will increase. So D sounds pretty good. And then pyruvate dehydrogenase is activated. Well, pyruvate dehydrogenase is that enzyme that controls pyruvate entry into the TCA cycle, pyruvate to TCA, acetyl-CoA to TCA. So this is an interesting answer, but it doesn't really answer why the electron transport chain is involved. So I'm going to guess answer D. And sure enough, there it is. Yeah, answer D. And that shows you the three steps that are inhibited by the NADH, and if the electron transport chain consumes those NADH, those three steps are activated.